Hello everyone, welcome to the Youth Research Institute podcast. My name is Harrison Xu, the founder of Youth Research Institute, and this is the show where me and some of my colleagues talk about recent scientific discoveries, review interesting research subjects, and interview researchers, professors, and graduate students about their research projects and how upcoming students can thrive in various research industries. Hello everyone, my name is Harrison Xu, and welcome to our second episode of the YRI podcast. Today we are covering a very interesting subject, something that I am kind of exposed to, but not this specific subject itself, and it is radiation oncology. I've been exposed to oncology before, so this general branch I am relatively familiar with, but radiation oncology specifically I've never heard of, or I didn't hear of before this interview that I had with Dr. Barcellos-Hoff. And Dr. Barcellos-Hoff is a professor at University of California, San Francisco, and her research is really interesting. And we talked a lot about it today, and I think it would be a really interesting interview for people to listen to. But before we actually get into the specifics and get into what we're going to talk about, let's talk about a bit of a timeline. So I'm going to spend the next 5 to 10 minutes just giving a bit of an introduction of what oncology is, what cancer is, how that actually works, how our therapy is being created for cancer, and then talk a bit about radiation oncology and why that makes sense and why it's effective. And once we talk about all that introductory material, we can actually go into the interview and actually see how we talk to Dr. Barcellos-Hoff and see what we actually discussed and what her research is. After that, we will just go on to our conclusion, just summing up the overall podcast, and then after that, we just end the podcast from there. So, let's just get straight into the nitty-gritty, which is, what is oncology? So oncology is generally the study of cancer. I'm sure many of you have heard of what cancer is colloquially, but knowing the science behind cancer will be quite interesting. Cancer is really cool because, and when I say cool, I mean it's really fascinating, but it's not a great thing, of course. It's not a good thing because it's a condition and it's a disease that has been causing millions and millions of deaths. But cancer is really fascinating because cancer occurs when a human cell mutates in some way, and this results in the over-replication of a cell. And I emphasize over-replication because it's not innately something that you think as really bad or something that's really deadly, but it actually really is. So in your body, a cell would typically replicate in response to a variety of things, like natural growth. If you're young, you naturally tend to replicate more because it leads to more growth and growing in height or growing in muscle, growing in mass. You can also have replication in response to stimuli that cause damage to your body. Like if you have a cut in your skin, your body tends to replicate to fill in that hole in your skin. And so your skin would be able to patch you up through cellular replication, otherwise known as mitosis. Now you might think if you have cancer cells, which are cells that grow uncontrollably, then theoretically that would be a good thing, because then that means like you could patch your cuts faster, or you can grow faster, and like you can become like Deadpool or something where you like regenerate really fast. But this is actually not the case, because cancer, when you over-replicate, causes masses to form in your body, and this is typically known as a tumor. Now these can be malignant or benign, which basically just means that they can either be really bad and super dangerous, or they can just not be very responsive and not really affect your life. But for malignant tumors, when they over-replicate, they continue over-replicating to the point that it becomes an issue, and it starts creating masses in your body that grow into and push into organs. And so this could worsen organ function. This could obviously be very deadly if it damages vital organs. It could also be that with so many cells that are being created because of this over-replication, you can't get rid of the cell waste that is being produced, and so it essentially poisons your body. Because something that you don't realize is that like you urinate so that you can get rid of waste in your body, but if you have a lot more cells being produced that are producing a lot of cell waste at once, it becomes much harder to get rid of that waste, and it can cause lots of physiological damage to your body. And also, cancers can grow into blood vessels or nerves and result in damage to those, and that can be very damaging to body function and can lead to internal bleeding and death. So yeah, it's pretty evident that cancer is really dangerous and can cause lots of deaths. But the issue now is how we treat cancer. The reason why treating cancer is such an issue is because cancerous cells in your body are still technically human cells. So this makes it difficult to treat cancer cells because if you, for example, ingest a drug or get a certain compound that will target and kill cancer cells, chances are those drugs are also going to attack other human cells, which includes healthy human cells. And this is specifically why cancer treatments often have so many side effects. For example, chemotherapy, which is a cancer treatment method where you use a variety of drugs to kill fast-growing cells, otherwise known as cancer cells. Although sometimes effective, oftentimes using chemotherapy will result in many debilitating side effects like vomiting or hair loss. And so cancer treatments are usually the things that cause a lot of these side effects that people have from cancer. Like, for example, usually cancer is not the thing that causes these symptoms of hair loss or vomiting that many people see happens to cancer patients nowadays, but rather it's the treatments that go behind it. It's the chemotherapy that people have to be treated with that actually cause these symptoms. Now, here's when radiation oncology comes in. So, oncology can be split into a variety of sub-branches, which are each based on their treatment of cancer. So, for example, there's medical oncology, surgical oncology, and the subject at hand, radiation oncology. Before this interview, I actually had never heard of radiation oncology, but the main things to know about it are that radiation oncology studies how radiation can cause cancer, but it also studies how radiation can be used to treat cancer as well. Now, something to be aware is that radiation does not necessarily mean nuclear power plants or like nuclear energy and like that kind of stuff. It's not like you're going to grow a second head, but radiation can also include a variety of things under the spectrum of electromagnetic radiation. Now, the electromagnetic spectrum is a spectrum of electromagnetic radiation. And so, under the category of electromagnetic radiation includes many things that many people are familiar with even if they don't realize it. For example, there is visible light. Now, this is just light that we can see. So everything that we can see is based off of visible light. So I can see my door, I can see my wall, I can see outside, the leaves are green. All of that is because of visible light. But there are also other types of electromagnetic radiation under the spectrum that we can't see. For example, there's infrared. If you ever had night vision goggles, then you can see infrared. You can see like the heat signatures off of people, and that's what infrared is. It mainly focuses on temperature. There's also microwaves, which are a type of electromagnetic radiation that is used in our kitchen appliance called the microwave. But there are also other types of electromagnetic radiation that is high energy and can cause damage to our DNA and cause mutations. For example, UV radiation. If you've ever worn sunscreen outside, then the purpose of that is to protect your skin from the damaging UV radiation from the sun. And if you let too much UV radiation hit your skin, then it can cause damage to your DNA, it can result in mutations and result in cancer in your skin. Another example are X-rays, which is a high-power type of ray that is used to essentially see your bones, right? This is like the thing that you see in hospitals where they try to scan if you have a fracture in your bone or something like that. X-rays are also really high energy and really useful, and they are another type of electromagnetic radiation. And so that's why radiation can cause cancer, but also it can treat cancer. This is very interesting and something I wasn't as aware of. So it's really fascinating to see how this subject has developed. And we will talk more about this, about how radiation can be used to treat, as well as how it can cause cancer in this interview. So, as you can see, radiation oncology is pretty cool. And now that we've covered a bit of this introductory material, let's actually get on with the interview.
Hello, everyone. Welcome to the Youth Research Institute podcast. I am your host, Ifan.
And I'm your host, Harrison, and we have a special guest today. And so, please introduce yourself, Dr. Barcellos-Hoff.
Hi, I'm Helen Barcellos-Hoff. I'm a PhD, basic scientist who studies cancer research at the University of California, San Francisco.
Yeah, thank you so much for being here. And I think we can just get started with just asking a bit about like your current research right now. What are some of the projects that you're currently working on?
Well, I am trained in two things. I actually got my degree in experimental pathology, which is the study of diseases. I did that at UCSF many decades ago. And then I did some post-doctoral training on cancer of the breast. And so right now, what I do is I study, I guess I should have said, and I'm a radiation biologist. So I'm a cancer biologist, a cell biologist, and a radiation biologist. So those are my three wheelhouses. And so what I study is how radiation acts to increase cancer in the population. So its role as a carcinogen that stimulates the development of cancer. And then the other half of my laboratory works on how we use radiation to treat cancer and how to improve the outcomes from that perspective.
Interesting. Okay, this is really cool. So do you have any specific projects that you're currently working on that is like especially interesting to you, or like what is your favorite project, I guess, that you're currently working on?
That's like asking you which one is your children is your favorite child. You can't pick. You can't pick. No, they're both interesting. I like so let me tell you a little bit about carcinogenesis, because that's not something that people generally think about. Is how does a tissue become a tumor? I actually used to have a billboard when I worked at Lawrence Berkeley National Lab, where they put my face up there with that little question, because essentially that's what I ask is how does a normal tissue, go through all the steps necessary for it to become a tumor, or for a tumor to rise in that tissue. And radiation can do many different things. The way it's generally characterized is that radiation causes DNA damage, and DNA damage, if misrepaired, cause mutations, and mutations cause cancer. I take a somewhat more holistic view to it, that radiation not only causes DNA damage, but changes the signaling between cells. Signals like growth factors and cytokines, and the way the cells communicate with each other is altered when they've been irradiated. And that is not a mutational mechanism, that's just changes in phenotype, not genotype, right? Not the sequence of DNA, but how the DNA is being used by the cell. And that to study that, what I do to look at those two different halves, right? The genotype versus the phenotype, is I've created a mouse model where we take out the mammary gland of the mouse at birth, at 3 weeks post-birth. And we can do that with a little surgery, very innocuous little surgery. And then at any point thereafter, I can transplant in a new piece of mammary gland, and will grow out and make a new duct. And so for me to study how radiation acts as a carcinogen, normally what I would do is just irradiate the mouse. I'd irradiate the little mammary gland in situ, inside the mouse, and just wait. But then I wouldn't be able to separate the effects on the DNA in the epithelium versus the effects on the cell signaling. So what I've done is I take out the epithelium, I irradiate the mouse, and then I transplant it with the mammary gland. And so that allows me to just look at how radiation has altered the entire mouse, if it has altered the mouse, in a way that affects the development of the mammary gland and its development of tumors. And what we found with this model, by using a little trick, we make the mammary gland that we transplant in very susceptible to making tumors by taking out a protein called p53. P53 is a brake on the cells, and so what it does is whenever the cell gets damaged, p53 steps in and says, okay, stop growing, or die, or repair your DNA. Well, if you take out p53, the damage occurs, but the response, stop, die, or repair, doesn't occur, so damage accumulates. And this kind of damage is what leads to cancer, right? So this kind of accumulative aberrations in the genome. So what we find is that when we've irradiated the mouse before transplanting with the p53 null epithelium, the spectrum of tumors changes. And I think that's been the most interesting thing we've learned. And we can actually show that what happens in my mouse model is similar to what happens in humans. In a particular population I'm interested in is women who have developed a second cancer after being treated for childhood cancers. So for somebody who develops Hodgkin's lymphoma, women who develop Hodgkin's lymphoma early in life, they get treated with radiation, cures their Hodgkin's lymphoma, which is good, right? But 20 years later, they have very high risk of developing breast cancer. And so I'm particularly interested in that population of people who are exposed to radiation for good reasons, right, to treat cancer. But how can we avoid the deleterious effects of a second cancer? So that's one study, and I think it's quite interesting. We've also used that same model to study radiation in outer space for NASA. There, we don't send the mice to outer space because we'd never get them back. But rather what we do is we irradiate them with the kinds of radiation that's found in space. So the space radiation environment is very different than that on Earth. It's full of these things called charged particles, which are the consequences of, nuclear fission in suns or stars. And so what it does is it generates very charged ions of molecules like iron or elements like iron or oxygen, and they're zinging through space. And along their trajectory, when they pass through your spacecraft, because they're very energetic. They're like, they will pass through the astronauts. And so the question is, well, what happens to the astronauts as a consequence of that radiation exposure, which we don't have on Earth? So we kind of recreate that kind of radiation exposure using big physics facility at Brookhaven National Laboratory at the end of Long Island in New York. We take our mice there and we irradiate them there and then do the same kind of experiment.
I see that. Okay, that's really fascinating. So I guess you just mainly talked about two main projects, right? So the mouse model where you essentially, so you irradiate the mouse without the mammary gland, and then you put a healthy mammary gland in and see how it grows.
Mm-hmm.
And you also talked about how you get rid of the p53 gene, which I think I've heard of before as well, which I think I've heard of it as like the savior angel gene or something like that, where it's like the last final gene, or like the final gene that will protect you from actually like getting cancer or like tumorigenesis. And so I see, so you get rid of the gene p53 and from the mouse model or from the mammary gland?
So it's only so you can knock out a gene in any gene in a mouse, right? So what I do is I take a mouse in which p53 has been knocked out, and I take out the mammary glands from that mouse, because that mouse will develop lots of tumors, and that's they die of lymphomas, and I don't want to study lymphomas. I want to study breast cancer. So I take out the mammary gland before they die of lymphomas, and I transplant it into a wild type mouse, a mouse that is normal and is matched in terms of the strain. So they're genetically similar enough that I can transplant that piece of tissue from the mammary gland of the p53 knockout mouse to a wild type mouse. That p53, a fragment of p53 null mammary gland will grow out and make a nice little ductal tree, which is what we call the mammary gland. It does look like a branching ductal structure, like a hand or a tree. And because it lacks p53, it has a high frequency of what we call transformation, which is that first step when the cell goes from being normal to being slightly aberrant. And slightly aberrant is evident by it might make more cells than it should, so it might proliferate more, right? Or the other thing that happens, which is quite evident to a pathologist, is that a normal ductal tree should look kind of smooth, and it'll get little growths on it, right? Like warts. These are early pre-neoplastic lesions. And so we can see that disruption of the growth organization by just looking at a slide. But what we really want to see is after a long period of time, it takes a year between the time I transplant to the time I find a tumor. During that whole time, all sorts of changes are occurring in the tissue. There's changes that are occurring in the local tissue near the malignant cells. There are changes occurring in the malignant cells. And then there's changes that occur systemically with the immunity, right? Because your immune system is supposed to get rid of all those bad cells. and it does a very good job of doing so. But eventually, for a tumor to be evident in the mouse, that means I can detect it by just rubbing its belly and feeling it as a little hard nodule, right? The immune system has to be compromised in some way, at least locally. And so all three of those things are going on continuously during the development of cancer. So I'm interested in how radiation perturbs those three things. And the only thing I've eliminated is the effect of radiation directly on the epithelial cells, but I know that radiation is acting on the tumor, on the tissue, changing the microenvironment. It's acting on the immune system, changing its ability to recognize the tumor. And as a consequence, when we reach the end of the year and we collect all these tumors, we can look at the spectrum of cancer, and we find that in an irradiated mouse, it's distinctly different than in a non-irradiated mouse, which supports my hypothesis that radiation acts through multiple mechanisms that are not genetic, at least not genetic at the level of the radiation-induced DNA damage, right?
Mm-hmm. Yeah, that's really cool. So you are using a lot of, I guess you're just using a lot of radiation therapy to see like how it affects different, I guess, aspects of like this mouse immune system. You said you talked about how it'll affect this tumor creation and like I guess tumor growth as well. And so I guess this kind of brings up another question that I had, which is how do you analyze, because you said throughout this year you look through like the molecular aspects of it as well as like the tumor, like how it grows and stuff like that, and you see how like the warts on this like ductal tree, like how they form. How do you like see all these things? How do you analyze all these things?
That's a good question. So there's a lot of different techniques we can use. Some of them require that we take a group of mice and we take them early before the tumor is developed. And what we can then do is we can look at the composition of the immune system. We can look at the circulating immune cells. We can look at the immune cells in the spleen, and we can look at the immune cells in the tissue. And we can do that by using a variety of different techniques. Some of them actually are just done on the tissue section, so we can stain them with antibodies, and then the antibodies localize specific proteins, and depending on the protein localization, we can say, oh, that's a macrophage, this is a lymphocyte, because this one has this protein called CD8 and this one has this protein called CD11B on it. And so we can look at the organization of those, we can look at the frequency of those. We can also take the tissues out, dissociate them into single cells, and then use another technique that also relies on antibodies. So antibodies are just a tool that we use. They're the same things that you generate when you have a cold or any kind of disease. It's your ability of your immune system to produce proteins that are specific to a particular piece of a protein. And so in science, for a lot of different reasons, we can take and inject the protein we're interested in, CD11B. We isolate that protein, purify it, and then inject it into a mouse. The mouse will make antibodies to it because it doesn't expect to see CD11B. So then we can purify those antibodies out, expand those, then we put a little essentially code a beacon on it, a fluorescent protein, and then we can detect when that antibody is stuck to our tissues of interest, our experimental tissues. So those are antibodies in a nutshell.
Yeah.
And so we use antibodies to either do it on tissue sections, right? So a slice of tissue where we can say, oh, the CD11B cells are right here, or they're right here, or they're all around this tumor. Or we can dissociate the tissue into single cells, and then we use a machine called a flow cytometer. We stain the cells in a tube, and then we run them through this flow cytometer, and it will count all the cells that are CD11B. So the great thing about a flow cytometer is you count thousands of cells in minutes, right? But you don't know where they were from. So we use both. For quantitation, flow cytometry is really great. For visualization, then we use microscopy and these fluorescently labeled antibodies to stain the tissue section itself.
Yeah, this is fascinating stuff. Yeah, because using antibodies is I'm pretty sure it's fairly common. I'd imagine kind of expensive because I think creating antibodies is pretty expensive, or like replicating that. But yeah, so like antibodies are essentially proteins that attach to a specific thing, right? Like an antigen is what the term is, and then so you're using antibodies that specifically bind to the protein that are unique to a certain type of cell, I would imagine.
Exactly,
right?
exactly. So we're looking for markers. So somebody else has characterized the cell and said, oh, these macrophages, they have, these 60 proteins on them. And somebody else goes in and says, okay, let's isolate the protein. Okay, since you don't want to do mouse on mouse, you take those proteins, inject them into a rabbit or chicken, any other species. You take the blood from those animals. You can just do routine kind of bleeds, take a bit of blood, just like you're donating blood. And from the blood, you can isolate the antibodies. There's a couple other fancier techniques to do that. In culture, you can do it in culture as well. And then there are other fancy ways of selecting for antibodies, but they're a tool. So now I have an antibody to a specific protein. I can identify that protein in different formats, on sections and in tissue, or in flow cytometry, or I could use it to isolate that protein. They're very widely used across biology.
Yeah, so yeah, antibodies to I guess you also would use antibodies that are like fluorescent, I would assume.
Well, we label them with the fluorescent moieties after we've isolated the antibody, right? So there's chemistry where you can attach the fluorescent, the fluorochrome, it can be any number of colors these days. And you attach it with just a little bit of chemistry to the protein, to the antibody.
Yeah, okay, that makes sense. And then you have an antibody that is like fluorescent, or like you have like some fluorescent moiety bound to the antibody, and then you basically just have the antibody on like that slice of tissue that you mentioned, or I guess if you mix it with or culture it with a bunch of cells, then it'll bind and it'll become fluorescent, and it allows people to see it, or it allows you to analyze it.
Right. Yeah.
That's fascinating. And also that makes a lot of sense, because I've also done a bit of like oncological research, and I have like dealt with similar things as well. So that's really cool. Yeah. And something else I did want to ask about is when you're talking about analyzing mice and like their slices of tissue, would achieving that, or like collecting these slices of tissues, or like collecting these cells, would that be like a fatal like operation, or are you able to keep the mice alive afterwards?
It depends on what, at some point, once they have a tumor, you have to, for humanitarian reasons, you can't let them go around with a big old tumor, so you have to euthanize them. But some of our experiments, you can do essentially what we do with people, we surgically remove the tumor so that the animal continues to live, right? And then we can analyze that tumor. Or you can take, or if you're interested in the blood, which cells are circulating in the blood is very informative in people, and we can do so similarly in mice as well. We can look at their blood cells and ask, well, are there more lymphocytes in this animal that has a tumor than in this animal that doesn't have a tumor?
Gotcha.
Different techniques, depends on the objective of the experiment.
Yeah, that makes sense. And then you also talked about your second project about how you're doing, like trying to simulate radiation in outer space, which is quite interesting. You talk about charged particles, so which you said were like, I guess, unique to outer space, or like more unique to outer space because they don't affect humans on Earth.
Well, this gets kind of a little complicated, but and remember, I'm a radiation biologist, not a physicist, but on Earth, you've heard of X-rays, right? You might have even heard of gamma rays. Gamma rays are naturally occurring, and they're kind of like X-rays. X-rays are man-made. So that's when you go in and have your teeth and they want to take an X-ray of your teeth, they use, a man-made machine to generate those, but gamma rays are comparable. And well, we call those types of radiation sparsely ionizing. And sparsely ionizing because that's what radiation is. Radiation is energy, right? And so energy interacts with matter, and that energy interaction can cause damage, oftentimes described as ionizations. So here's the biologist view of a low LED, we call these different things. I get them mixed up sometimes, but not mixed up, but I use them interchangeably. Sparsely ionizing radiation is like a BB gun. The ionizations occur as a function of the distance from the source, right? The closer you are, the more ionizations there are, just because, but they're like a BB gun. The ionizations are just random, right? In that volume of tissue that you're irradiating, or it could be anything that you're irradiating, the ionizations are just going to be random. And they're going to be sprinkled as a function of the amount of energy you have and the distance from the source. Okay, so that's what we call, that's the kind of radiation we're usually exposed to on Earth. In contrast, in outer space, there's that kind of radiation, sparsely ionizing, but there's also something that's called densely ionizing radiation, which are these particles that are accelerated nearly to the speed of light by this nuclear fission, right? And so those particles like iron or oxygen or gold, anything in the, essentially in the element table, can be accelerated nearly the speed of light. I describe them like cannonballs. So along the particle track, now there's a single particle track, right? And along that track, the particle is going like this, all the ionizations are occurring right there, right along that track. And so we call it densely ionizing because although it's not distributed like the ones I was talking about, all the ionizations occur along the particle track. And so you get this very micro dosing of the radiation in that tiny little area around the particle. Now, of course, you could have a lot of particles, boom, 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 right? So you could have a lot more dose, but the kind of damage is quite distinct. And when we think about it in terms of DNA, we think about it being clustered. So you'll get lots of double strand breaks of the DNA all in one area, which makes it very hard for the cell to repair. In contrast, you take the same amount of energy, but you use from sparsely ionizing radiation like X-rays or gamma rays, you're going to have the ionizations distributed in the nucleus of the cell. And so it's easier for them to repair, right? So it's well, I don't know if that's right or wrong, but that's the way it is, right? So densely ionized radiation occurs outside the in outer space. On Earth, we're protected from that by the magnetosphere, which surrounds the Earth and essentially deflects many of these ions. Some of them make it through, and so you get actually more cosmic radiation when you fly because you're up high and you don't have as much atmosphere. Still have the magnetosphere, but you don't have as much atmosphere. That's one source of cosmic radiation. If you live in Denver, you get more cosmic radiation than if you live in Phoenix, just because the altitude. And I guess the only other interesting thing I can tell you about densely ionizing radiation is that's what the aurora borealis is due to.
Oh, that's cool.
Yeah. So the northern lights are evidence of densely ionizing radiation. When you have these solar flares, they spew out a lot of these densely ionizing particles, and that's what we're seeing when we're seeing the northern lights.
I see. So you're essentially testing how densely ionizing particles can affect tumor creation or like carcinogenesis.
Right, because we don't have any people who've been exposed to this. So what is NASA going to do? They know the space radiation environment is different. We know that a 3-year mission to Mars will the astronauts will be exposed to well, it's kind of hard to, it's a unit of dose called a gray. A unit of radiation is called a gray. There's also called other things, so it's very confusing. But so that's a relatively large dose, but more concerning but it's spread out over 3 years, so less concerning. But then it's this densely ionizing radiation, so we don't know very much about that. So NASA funded a lot of researchers to study densely ionizing radiation generated at the alternating gradient synchrotron at Brookhaven National Lab, where we took and put our mice in the beam and said, okay, now let's do our experiments and see how carcinogenic it is. It's considered to be more carcinogenic than sparsely ionizing radiation, at least in our mouse models.
Yeah, that makes sense, and that's really fascinating, and I'd imagine also has a lot of practical applications because, of course, if you're able to study how densely ionizing particles can affect carcinogenesis, then you could also probably create some form of protection against it for like astronauts or other people going into space.
Well, that is actually where NASA has decided to put most of its effort. So they got it kind of decided, well, we know, okay, it doesn't look good. It looks like it's more carcinogenic. So what can we do about that? So those in NASA language are called countermeasures. And their countermeasures are something that you can give to the astronaut that are relatively innocuous, that would prevent them from developing cancer once they went to Mars, came back, and 10 years later, 20 years later, right?
Yeah, that makes sense. Especially since like with the magnitude of distance between like planets or like just going into space and having these missions, I would imagine it would expose them to a lot of radiation. So it's very key to have some form of protection. So yeah, that's super interesting. That's really fascinating. And so those are like the two main projects you talked about, right? So the mice, sorry, what was it? Treating
different kinds of carcinogenesis with different kinds of radiation. So what we call radiation quality. So densely, sparsely ionizing radiation, we've done experiments with those. And then the other half of the lab studies radiation as a therapeutic. So when you get cancer, radiation is used in more than 50% of all patients to control the tumor or eliminate it, right? So there, you're not thinking about the risk of radiation, but rather the benefit of radiation is that because it causes DNA damage, it kills tumor cells. And so you get rid of the tumor cells, and that provides a significant benefit for many patients.
Yeah, and that makes sense. Although I do have a question about radiation oncology, actually, or like radiation therapy, which is like, is there a way to selectively target areas of a human body with like radiation therapy? Because I know, especially with cancer therapies in general, like just chemotherapy, for example, which is, if people don't know, is just like treatment of cancer through like ingestion of a variety of drugs or like a combination of chemotherapeutic drugs. I know that obviously has lots of pretty painful side effects because of the fact that it will also target healthy cells. And obviously this is like a huge dilemma in cancer or like oncology and like treating cancer, because cancer cells are technically still human cells, or like they derive from human cells. Yeah, so like if you want to target a cancer cell, it also targets human cells. So yeah, I wonder, is there a way to like selectively target cancer cells in radiation therapy?
Right. So actually that's the greatest advantage of radiation, because it's physics, and radiation oncology rests upon a very strong bed of physics and physicists. Physicists can figure out how to target the beam, the radiation beam that I've been using my fingers to demonstrate here, but you can use a lot of different techniques to localize most of the energy into the tumor volume. So we use a variety of different things. First of all, the patient tumor is imaged, so they know where the tumor is, right? And then they make a treatment plan, and they pick the radiation delivery mode, because there's different kinds of radiation radiotherapy machines. They pick the mode that is most suitable for the location of that particular tumor. Sometimes you have superficial tumors very close to the skin. Sometimes you have deep-seated tumors. And so what you're trying to do with the physician and the physicist who work together to create the treatment plan use is the physical properties of the radiation and the medical knowledge of the physician to say, oh, well, we want to deliver this much dose to that tumor, but we don't want to, like you were saying, we don't want to spread it around. We want to get as much as possible into the tumor and spare the normal tissue, right? So that's what you're talking about. Chemotherapy, you take systemically, it's like an aspirin. You may be taking aspirin for your head, but it affects your knees and everything else because it's circulating. And so chemotherapies are a broad class of drugs that are selectively toxic to cancer cells. But that selection it's not like the normal cells are protected because they're normal. It's just that they're better able to repair the damage that the chemotherapy does to all the cells in the body. So that's why chemotherapy can be very debilitating. You lose your hair. You can lose your hair from radiation too, but only for certain kinds of radiation treatment. But if you have lung cancer, you can go in and with the radiation specifically target that lung nodule so that most of the dose is in the tumor. You still get some dose around the tumor and the rest of the lung, but it's much, much less.
I see. So it's a lot more selective compared to like therapeutic drugs.
Yeah, I always like to say it's the original targeted therapy. Targeted therapies are cancer therapies that try to take advantage of the fact that cancers have mutations, and so we design drugs that will specifically kill the cells that have a particular kind of mutation, right? So that's called targeted therapy. But radiation has always been targeted because you can always deliver the dose generally to the site of damage of the tumor.
I see. That's really cool. And so for radiation therapy, how does it exactly work? Like I know you shoot like high power beams or like some form of radiation into the tumor cell, but does it like destroy the DNA? That's what kills the tumor cell, or like does it destroy the cell membrane? What's going on?
Well, we think that the lethal lesion with ionizing radiation is DNA damage, so a double strand break. So your helix, I can't do a helix with my hand, but the double helix that the DNA is formed has two strands of the DNA wrapped around each other, and so ionizing radiation can break both strands, and that is hard to repair for the cell, and so cells oftentimes die as a consequence of that. And cancer cells are less proficient in repairing their DNA, so we can give that DNA damaging radiation to the tumor and kill those tumor cells, and then give it again, kill more tumor cells, give it again and kill more tumor cells until we eliminate the tumor, hopefully, right?
Yeah, gotcha. Okay. So that's really interesting. So yeah, a lot of your research is based on, I guess, like two main branches, which is looking at how radiation can cause cancer and how radiation can essentially cure cancer.
Exactly.
So that's kind of like a double-edged sword. That's pretty interesting. And I guess like I learned a lot about like the selectivity of radiation therapy. So I think now we can move on to a bit more of like a student-based part of the interview. So just asking like, well, I guess like just to start off, like how did you get into the industry or field of radiation oncology?
Well, yes, I think that's always interesting to ask how people get into a particular field, right? So I knew I wanted to be a scientist when I was in college. Did I know that in high school? I might have thought about it. I was thinking when I first went into college, I was more in psychology, kind of social science. But I got to college and I decided, no, I'm going to go in science. And actually, I had I had a job, a work-study job, dissecting brains out of snakes, and trying to understand the neuroanatomy, how the neurons are connected in the snake brains, where I learned to handle snakes, which I don't want to do anymore, but it was an interesting thing to talk about over, beer and wine at the college parties. Oh, what do you do? I took out a couple snake brains today. And so that was interesting way to getting into science, because I learned kind of processing of tissues, and I learned to study cell-cell interactions. I took biology courses. When I finished college, I had decided I wanted to be a marine biologist. And I came to San Francisco thinking that, oh, well, this is a good place. I'm near an ocean. I can, do marine biology here. So first thing I had to do, of course, is make enough money to go to college, to graduate school. So I started looking for a job, and took me 3 months to find a job. And during that 3 months, I volunteered at the Oceanic Society, where 3 things happened. I found out that there were a lot of unemployed marine biologists. Number 1. Number 2, I found out that I got terribly, terribly seasick.
Oh,
when I got on a boat, which was not a good, strategy for marine biologists. And then number 3, when I got my job, I actually started a job at the Brain Tumor Research Center at UCSF, using my vast knowledge of snake brains, right? So then I realized actually any problem can be interesting if you get enough information about it, right? And so I became very interested in brain tumors, and I ended up doing my graduate work there in the brain tumor research, where I studied how radiation acts as a therapeutic in brain tumors. So I did a lot of studies there. To understand better the DNA damage response and how to improve it, I make the cancer cells die better, more. So one of the things you try to do when you're in biology, or when you go from graduate school to postdoc, right, after your doctorate, is you try to learn different skills. And I decided that one of the things I found so interesting in brain is how cells were interacting through this extracellular space called the extracellular matrix. So extracellular matrix is the stuff like collagen that you hear about all the time when you're listening to skin commercials, right? Collagen, you've got to have better collagen, right? There are lots of other extracellular matrix proteins, but I got very interested in extracellular matrix. So I went over to Lawrence Berkeley lab, and I decided to study extracellular matrix. So I studied brain tumors in rats. And so I went over and studied extracellular matrix in mouse mammary gland, where we studied how the matrix, that the proteins the cells sit on, actually helps them to function and tells them essentially what to be doing. So I had 2 very different perspectives, brain tumors and mouse milk, because that's what the mammary gland does, right? It makes milk. So that's the product. And when I finished that, I had the opportunity to, start my own laboratory. I decided I wanted to study how radiation acts on the extracellular matrix to make cancer. And that's how I ended up doing that whole sequence. It's sometimes when people go into science, they think, oh, I'm going to study this one particular problem because I have a family member who's afflicted by this. I think there's also a large group of scientists who just, it's kind of serendipity what they are exposed to, and they just have a mind that wants to think about these kind of problems, and they'll take anything. I could probably still go back to marine biology and study something that doesn't require boats, maybe something on the seashore, right? But it just happened that I really enjoyed that group. Yeah,
and I think that's like a really inspiring story, I guess, because a lot of people are like worried about having to have like a job or like their life planned out immediately, and it goes to show like you don't have to plan it. Like you can just figure it out along the way, and like you might change industries, change what you think. It's just whatever you find interesting. So that's really cool.
I think it's really important. One of the things that I always ask my students when they come into the lab, what kind of data do you like to think about? Okay, so scientists like to think about data, but there's different kinds of data. I've described imaging data, right? Quantitative data. Some people are very pattern oriented. Like I'm trained as a pathologist, so I can. It takes advantage of the fact that I'm very good at recognizing patterns. I have a very good visual memory. Some people all biologists have to have a very good memory, period, right? You really do, because you have to remember all those little details, because there's no, it's not like physicists. I've had physicists in the laboratory, and they say, this is so hard. If I forget something, I just rederive it. You can't do that if you're a biologist, because there are no first principles. I mean, you can so biologists tend to have good memories, but they can like quantitative data, they can like image data, they can like the way your mind works, the kind of information that makes your head, really puzzled, that you think about it and you find it interesting to think about. If you figure that out early in your life, test it out, do I like to do lab work? Do I like to work with numbers? Do I like physicians talk about this all the time. They like to talk to people. Okay, scientists are okay talking with people, but physicians like to talk to people, right? And so there's a lot of things that you can figure out about yourself that will help you identify something that you'll be happy in the long run. Because if you can be gratified with your work, and you're not doing it because you think it's, the way to make the most money, or it's something that your great-grandfather always wanted you to do. If you instead think about, what makes your brain work well, you'll be happier in the long run. It could be numbers, it could be writing. Scientists actually do a hell of a lot of writing. So it's good to practice, like you had suggested with your magazine, writing things out. We do a lot of writing. Some people, I get in the laboratory, they say, oh gosh, I really love science, but I hate writing. I go, well, we're gonna have to find you a slightly different career path. Because as an academic, I write a lot.
Yeah, and it's also just really great to know, especially as like the Youth Research Institute, we're trying to expose students to as many experiences, or like hear from other people as many like different things about like what would it be like to follow this kind of career, and hearing about like the various paths, right? Like there are also like many things that you might think aren't that useful in STEM, like writing is surprisingly really useful, or reading literature for like your studies, and like being aware of like the different types of like thought processes for different types of scientists, like you mentioned, like physicians or physicists versus like biologists. All these like differentiations are very key. And Yifan, did you want to say something?
Yeah. So I guess you talked about how you didn't really know what you were wanting to go into in high school and college, right? But then once you're in this industry of oncology, I was wondering if there's anything that you want to tell the younger students, like students in middle school, high school, that wants to go into this field, if there's any like advice or tips that you could give to them on what they should do, what they could do, and like what activities would benefit them in the long run.
I think the things that you just pointed out, like getting exposure and finding out which things you like to do. They're actually, here's, I guess, one of the things you may not be quite as aware of, but maybe a little bit more so than most other people. All of science is taught by essentially a continuum of scientists. I went into the laboratory, somebody taught me how to dissect the mice, I taught my group how to do the dissection of the mice. Everybody teaches each other. So it is really one of the few areas I know of where you really do an apprenticeship with, and you learn hands-on, because there's lots of idiosyncratic techniques in like taking out mammary glands, How many people know how to do that? My children know how to do it because they had to watch me do it while they were growing up, but they didn't become scientists, by the way. So I think getting exposure, like you said, to different kinds of experience, thinking about what kind of things, do you like to be out in the open? I often ask radiation oncologists, do you realize when they come in for training, because all radiation machines are in the basement because they're heavy. I've asked residents who are applying for positions in training, I said, do you realize you're going to spend the rest of your life in the dark, in a basement, with no windows? It's something to think about. That could drive some people, kind of crazy. The other thing, so everybody learns by asking. Everybody learns by example. We don't have, we have comprehensive courses that teach you about techniques, but actually doing the techniques you learn hand-on. So it's great to do an internship if you can, but if you can't, don't feel like that's the end all and be all. The other thing is that scientists, because we've all learned the same way, are generally pretty open to training others. There's certain limitations, high school students at UCSF, you have to be over 16 before you can be in a laboratory. It's just a safety rule. Okay, so that's one thing we've learned. But you can. I've had several high school students in my laboratory. Some have gone on to do their MDs and PhDs, I'm very excited to say. So you have to ask. My criteria is you have to be stubborn and persistent, because while we've all learned this way, we don't always have room. We don't always have somebody to, room meaning that, physical space, but we also have to have somebody who trains you, right? So if we have only 5 people in the laboratory, I can't take 5 students because everybody would be busy with students, right? So I think it pays to be persistent. It pays to, if you want an internship, really should start in January or December and just send out lots of emails. Eventually somebody will write back. Some people don't. I always try to say, sorry, not this year, but you can always look. And then there are several programs, and UCSF has actually a number of programs for high school students as well as for college students. So again, you don't have to have it in high school. You could do it in college, perfectly fine. It's good to get an idea of whether you like bench science. There are other kinds of science. There's bioinformatics, fascinating area. Lots of informatics, lots of computational studies in biology now. So those are all good things. So I would say, don't be shy, ask, be persistent though. Don't be put off when people don't answer your emails. They're busy, and eventually something will hit. Use the programs, right, that are out there, contact them. And in the absence of any of those getting you an internship, just read and explore the literature. One of the ways that you can go make a connection to a laboratory is just asking about their paper,
Yeah, so I completely agree with you. And regarding like the finding opportunities and finding research opportunities through cold emails, I've done that personally. And I think one of our goals for our organization is to streamline that process by reaching out to professors and then connecting them with our students, right? So I guess before we end, I do want to ask if you could link some of the opportunities that UCSF has for high school and college students. If you could link them to us after the podcast, that would be great. And then we could share that with our students.
Yeah, absolutely.
Yeah, and I think that is coming to the end of our time. So thank you so much for talking to us, Dr. Barcellos-Hoff. This was a really great and informational talk. We learned a lot about radiation oncology, and it's just really fascinating to see the different studies that you're doing and like the kind of, I mean, I guess like there's outer space studies as well as like on Earth. It's just, I guess, out of this world. It's super cool. So yeah, thank you so much for talking to us.
Well, it was a pleasure. I always like the opportunity. Oh, I forgot to use my favorite term for NASA work, intergalactic cosmic rays, right? So we get to study intergalactic cosmic rays. So it's a great profession. It's a great career. So good luck to you all.
Yeah. Thank you so much. Have a good day.
You too.
Thanks.
And that was our interview with Dr. Barcellos-Hoff. Thank you again so much, Dr. Barcellos-Hoff, for coming over and spending your time to talk to us about your profession. And I think this interview was really informational, and it's really fascinating for any biology buffs out there, because cancer biology is super expansive, and I think cancer in general is a really fascinating subject and kind of contradictory, I guess, in a sense, because of the fact that it's so researched and there's so many different treatments, but yet there's not really that good of a cure, or like that permanent of a cure, and it's just really difficult in general to treat. So learning about radiation oncology and radiation treatments is super fascinating. And seeing how Dr. Barcellos-Hoff's research is focused on learning how radiation causes cancer, as well as how radiation can be used to treat cancer, it's really fascinating to see the two different directions you can go with a radiation oncology. And additionally, she even goes into cancer biology in space and using, she says, intergalactic cosmic rays, and at least studying those and their influence on cancer development. And I think her research is super fascinating. It might be really dangerous. I imagine it really is, but it's super fascinating. And of course, it's really, really useful to our lives, because many people do get diagnosed with cancer, unfortunately. And so figuring out ways to treat it effectively is really key. That was the entire podcast episode of Radiation Oncology. I hope you all have learned a lot today, because I know I have. It has been an entire roller coaster of knowledge, learning about radiation oncology and just oncology in general. And so learning about Dr. Barcellos-Hoff's research, it's mind-blowing. And I hope that everyone here has also learned a lot and found this very interesting, and as interesting as I did. But anyways, I hope to see you all next week for our next episode, and we will be interviewing another scientist or another researcher from a college or professor, and we'll see how their research has been applied in the real world and what they do in their research. See you guys later. To learn more about the Youth Research Institute and get involved, check out our website, www.youthresearchinstitute.org, and subscribe to our newsletter to become a member. You can also keep up with our content on our Instagram at Youth Research Institute. Thank you all for listening, and we'll see you next week.